Hello, hello everyone. Happy New Year. Welcome to our Molecular Connectivity online series, or MCOS. My name is Cindy from New Jersey Institute of Technology. For those who are coming here for the first time, the Molecular Connectivity Working Group is a community of researchers dedicated to promote molecular imaging as a tool to study brain connectivity. We plan this platform to exchange ideas and promote the latest research on molecular connectivity. You can go to our website, subscribe to our newsletter, and follow us on X and or LinkedIn. Before we start, just a quick note. We are recording this session and we'll upload this to our YouTube channel. So we ask you to not record it privately or upload it to any third-party platforms. So now I'm pleased to introduce our today's speaker, Dr. Sarah Genon. Dr. Genon is a cognitive neuroscientist specializing in neuroimaging, machine learning, and brain behavior relationships. She is a Hessenberg professor at Heinrich University Dusseldorf and a group leader at Fortune Jülich in Germany. Today, she will presenting on connectivity-based parcellation to map brain organizations. Thank you again for joining us, and let's welcome Dr. Genon.
Thank you very much for the nice introduction. So I will share my screen. This should be the right one, right? You can see it. And
Yes.
we'll maybe get a pointer too. It will help. Okay. So yeah, thank you very much for the invitation. I'm really happy today to present our work on brain parcellation. So I will start with some conceptual consideration about why we do brain parcellation. And then I will focus more on connectivity, on metabolic connectivity-based parcellation, as this is of interest for this particular group. And all along this talk, I will focus on the hippocampus because this is a very important region in many psychiatric and neurological disorders. And this is a studied region that I've studied a lot with parcellation technique. So I would like to start this topic by drawing an analogy between brain maps and geographical maps. So if we are interested in studying a particular brain region, some phenomena in association to this region, for example, the hippocampus, which is a very tiny region in the brain, but this may worth investigating in relation to many phenomena. This may be the same way as we might be interested in study a specific country, such as Belgium, which is a very small country, in association to some phenomena. So let's say we want to study behavioral habits in the Belgian population. So we need a map of Belgium, and there are many different maps that exist, official maps of Belgium. So for example, we can have a subdivision based on geological features. So these are features that characterize the soil, the ground. So we have regions that are more sandish, some regions that are more stonish, I would say. And this map is relatively aligned with the map of provinces in Belgium, as you can see. But there is no perfect fit. So it's partly aligned, but not fully aligned. And I would say that none of these two maps would be relevant for studying behavioral habits in the population or behavioral traits. Why? Because behavior is highly influenced by culture, which is itself highly influenced by language. So for me, if I would study behavioral habits in the Belgian population, I would probably look at the map, use the map which is based on language communities. So there is the Dutch-speaking communities, the French-speaking communities, but also the German-speaking community in Belgium, which is an official community for those who might not be aware of that in Belgium. So that would be for me the most appropriate map. And this partly aligns with the official map of region, which differentiate the Flanders from Wallonia and from Brussels. But as you can see, it's not a perfect matching neither, because the maps of region mix together within Wallonia people who speak German and therefore have a Germanic culture with people who speak French and therefore have a really French culture. And so I would say that the latter map wouldn't be the perfect map for studying behavioral habits among the Belgian population. So I hope that with this example, you see why it is really important to think what is the most relevant map or subdivision or territorial representation to study a specific phenomenon. So now coming back to the brain, how do we parcellate the brain? So we use brain parcellation. This is actually an umbrella term that covers a wide range of techniques. I wouldn't go into the detail of those techniques here because this could be a whole day course. But basically, we can differentiate two main techniques. So we have boundary mapping approach. So this is what is typically used or was typically used in cytoarchitecture. For example, the idea is to identify when there is an abrupt change in the feature profile. For example, if we go along the surface of the cortex, indicating a border between two areas. So this has been used a lot in cytoarchitecture, as I said. But later, when we start using neuroimaging data, mainly from MRI scanner, we had whole brain data, and this looked more like a multivariate patterns, and therefore clusterization and factorization approaches play a bigger role. That being said, there is no specific requirement for using one technique for one type of brain markers or brain features. So for example, boundary mapping has been used traditionally with cytoarchitecture data, but this has also been used with resting state functional connectivity data, for example. And clustering factorization is used a lot for connectivity features, but it has also been used with microstructural features in the big brain, for example. All right. So when we have parcellation technique, how do we draw map and what kind of map can we have? I will focus here on the hippocampus because, as I said, this is a region in the brain which I have studied a lot. So this is deep in the brain. You can see it here. I will move the pointer so you see it well. And a few years ago, when I started reviewing parcellation studies on the hippocampus, there were already a lot. And as you can see, we have a wide range of partitions. So they can be really different from one study to the other. And what I did here is that I have ordered those different partitions based on the type of brain markers or brain features that were used. So on the extreme left, we have studies that focus on local tissues properties, such as those derived from MRI, like gray matter volume, or FA or MD, and as well as features derived from histological materials such as available in the big brain. And on the right side, on the extreme right, we have partitions that are based on features that reflect large-scale functional integration. So this is typically connectivity features. And as you can see, the pattern that we have can vary a lot across those different parcellation based on the features that they use. There is actually some general pattern that emerge there if you haven't noticed it. So the basic idea is that if we look at local microstructure, like, for example, cytoarchitecture, as in the work done by Catherine Ammons in 2006, we have a differentiation of the hippocampus into different fields or subfields, which are arranged along the medial lateral axis. So you can see that this goes from medial to lateral, and these fields are also arranged along inferior superior axis, but we cannot see it there. But this contrasts very much with what we have when we look at connectivity markers. So when we use connectivity markers, such as functional connectivity, we typically have an organization along the anterior-posterior axis. So you can see it here. The most famous model is a subdivision of the hippocampus between the head, body, and tail. And we know that these functional subdivision are highly relevant for studying behavior. We have shown across a range of studies that the anterior part has differential behavioral involvement than the posterior part. So now how do we get those connectivity-based maps? The idea is quite simple. So whatever type of connectivity measure you can have from an MRI or a PET scanner, if you are interested in a specific brain region, such as here the hippocampus, we take the voxel within this region and we look at their connectivity profile across the brain. So with which region they tend to functionally interact, for example, based on functional connectivity. If we take these two guys, we see that they have different pattern of functional connectivity across the brain. So we then just map the connectivity profile of all voxel and we cluster them based on the similarity or the differences in their connectivity profile. So for example, here we differentiate anterior versus posterior region in the hippocampus. And this idea actually works for any kind of connectivity features. So typically this has been used a lot with resting state functional connectivity data. This has also been used a lot with task co-activation derived from meta-analytic connectivity modeling. This has also been used with structural connectivity feature derived from diffusion, so probing white matter tracks. And this has also been used with structural covariance. So structural covariance is, for example, covariance based on gray matter volume. So region that, so we look at region in which gray matter vary across subject. But structural covariance is a bit of an hybrid method for me because it probes into both functional integration as well as local tissues properties, because regions that, for example, tend to be co-atrophied in aging or co-maturate across development would tend to be involved within similar behavioral system as they work together functionally. But they will also be tend to have similar microstructural profile. So in that way, structural covariance is a bit hybrid, is probing both functional integration and local tissue properties. Now the question is, what would we have if we use metabolic connectivity? So this is something we got interested into a few years ago. And we, of course, focus on the human hippocampus. So we aim to map the metabolic connectivity based subdivision of the hippocampus. So for that, we went to the ADNI dataset. We selected 266 FDG PET scan from cognitively healthy participants. We use Clinica for, so the Clinica software for pre-processing. We use standard pre-processing. We normalize metabolism by the pons. And we derive metabolic connectivity between each hippocampal voxel and 464 brain parcel using Pearson correlation. Then we perform clustering using a k-means algorithm, and we evaluate stability of the clustering using a bootstrapping approach. Why is it important to assess stability? This is because clustering is typically an unsupervised machine learning methods, meaning that we don't know what is the truth, and therefore we can subdivide any region into 2 cluster, 3 cluster, 4 cluster, 5 cluster, and so on. So we need a way to decide which partition, which level of subdivision to focus on. And to do so, here we decided to focus on stability across resampling. And we could see that the subdivision that showed the highest stability across resampling was this 5 cluster solution. This is also the subdivision that portray a symmetrical subdivision, so between the left and the right hippocampus. So this is how it looked like. So here, this is a view from the top, a superior view. And here, this is an inferior view. So you should think that we have take the brain and we have pivoted so that we can see the inferior part from the inferior part. And we can really see here that the subiculum that goes all along is subdivided into an anterior and posterior region. So this parcellation really subdivides the hippocampus between anterior and posterior regions. So we have an anterior subiculum here in red, a posterior subiculum in blue, and we also have an anterior CA here in yellow and a posterior CA here in the tail of the hippocampus. So this parcellation is based on the way the voxels, the profile of the metabolic connectivity of the voxel across the brain. So the question is of course, then what differentiate those regions in terms of the metabolic network? So we map that for each region and we look at the specific network, molecular network for each subregion. And what we see is really differences between the anterior and the posterior subiculum. So we can see here that the anterior subiculum is more specifically connected to a lateral temporal region as well as a prefrontal region, while the posterior subiculum has an extended network across the brains, mainly covering medial part, but also lateral frontal part, as well as IPS, interproparietal junction. And this is, I should say, this is really typical from the posterior subiculum. Across studies, we really see that this is a core subregion within the hippocampus that connects the hippocampus to the whole brain. It really integrates the hippocampus into a functional brain system. So this is really a core region. In contrast, the intermediate pink part here is mainly local connectivity, so within the hippocampus. The anterior CA in yellow is mostly connected to amygdala, and the posterior CA is mainly connected to the thalamus preferentially, because I'm talking here about specific connectivity of each subregion. And when I saw this pattern of metabolic networks for each subregion, this actually remind me of the structural connectivity, so the white matter tract in the brain. So here I put the white matter tracks from different studies that have been estimated from diffusion. And when I look at the connectivity of the anterior subiculum, this reminds me of the uncinate fasciculus, while the posterior subiculum has a connectivity that reminds me of the cingulate bundles here in blue. And finally, the posterior CA has a metabolic connectivity which reminds me of the fornix. So this would argue for the neurobiological relevance of this parcellation. And these networks also have slightly different behavioral involvement. So we perform some kind of behavioral decoding across activation studies of each metabolic network. And basically the network of the posterior subiculum is associated with a range of high-level cognitive function. So this fit with the connectivity profile of the subiculum, which is really extended, as I say, across the brain. And this is in order to observe all these high-level cognitive functions, such as working memory, episodic memory, and a highly demanding, cognitively demanding task. In contrast, the anterior subiculum was associated more with object recognition. I mean, the network of the anterior subiculum was more associated with object recognition function. And of course, the amygdala, which is connected to the anterior CA mainly, was engaged in emotional processing. So we can really see that we have subregions that have relatively specific metabolic connectivity associated with some specific or more specific, more preferential behavioral engagement. But then the next question you may ask is actually this parcellation relevant for studying brain metabolism? So to look at this question, what we did is that we took a healthy set of subjects and we look at pairwise differences in local brain metabolism. And Actually, we had significant differences between all pairs of region within the left and within the right hippocampus. So you can see it here. We have different metabolism between the subiculum and the CA region, but also within each field, we also have significant metabolic difference between the anterior and the posterior subregion. So typically, the metabolism is higher in anterior region than in posterior region, and both for the subiculum and for the CA, and both for the left and for the right hippocampus. So this would argue that this parcellation is really relevant for studying brain metabolism. And to go even one step further, we have also looked at an anatomical parcellation. So we took the segmentation, which is provided by FreeSurfer, which is a very robust and well-derived segmentation, but this is anatomical. So this follows cytoarchitecture work, and accordingly, we have different CA field, and we have the subiculum, which cover, as I said, the inferior surface of the hippocampus. You can see here, and it's a whole region, so it's not subdivided like in our parcellation between the anterior and posterior subiculum. And accordingly, when we compare brain metabolism between each pair of hippocampal segment derived from FreeSurfer, we had a lot of non-significant differences. So basically, this FreeSurfer based segmentation is not very much sensitive or adequate to study brain metabolism derived with FDG PET. Instead, our parcellation really have these differences between anterior and posterior parts, so it doesn't neglect this difference between anterior and posterior. And so then the next question was also, is this anterior-posterior differentiation also potentially relevant for studying dementia, so Alzheimer's disease? So what we did to look at that question is that we computed within each individual, within each subject, a delta between anterior and posterior subregions. So we had a delta for the subiculum and a delta for the CA, for the left and right hippocampus. And so this delta basically reflects the imbalance between the anterior and the posterior hippocampus. And what we saw is that this delta, this imbalance, this discrepancy in brain metabolism between the anterior and posterior hippocampus was maximal at the late MCI stage. So this is the stage at which patients are closer to Alzheimer's disease. They have stronger memory deficit than early MCI, but they are not yet Alzheimer's disease patient because they do not fulfill the criteria for dementia. So this is really interesting that we have this discrepancy in LC older people, but this discrepancy, this imbalance also change in mild cognitive impairment and might be a relevant biomarker actually in Alzheimer's disease. And so you should think if we just take the whole subiculum or the whole hippocampus or the whole CA in which we have this difference between anterior and posterior region, what you do is you average between the anterior and posterior regions. So you end up with this, which somehow you lose significant information by taking this average across the whole hippocampus and ignoring this difference between anterior and posterior region. So to sum up, I hope across this talk I could convince you that different brain parcellation reflect the complexity of brain organization. And I also showed that metabolic connectivity-based parcellation of the hippocampus portray both an anterior-posterior differentiation as well as a fields-like differentiation, which we know from cytoarchitecture. It captured differences in local metabolism across the hippocampus in healthy people, but it also captured Alzheimer's disease-related changes in brain metabolism, So I would conclude that metabolic connectivity-based parcellation can provide relevant maps to study brain metabolism in healthy and clinical population. So my lab worked a lot on this brain mapping question, and I would like to switch to a project that we are doing at the moment. So what we aim to do is that we would like to build a normative model of brain metabolism. So this means that for each region of the brain, we would like to know what would be expected in a healthy population for a certain age and a certain sex, and possibly also a certain level of education, in order to better identify deviance in clinical population. And at the moment, we have built normative model based using deep learning approaches, which require a lot of data. And we have mainly data from the US. So we have normative models such as ADNI. So we have normative models which are very much US focused, and we would like normative models which are representative of the whole population, not only the US. And therefore, we are looking for people who would like to share data through a collaboration. So that would be through association to our work. And we are looking for healthy people. We define healthy at baseline, so cognitively healthy, and we need some evidence that those people are not going to develop Alzheimer's disease. So this could be either a follow-up or either some biomarker for Alzheimer which are negative. And of course, we need some basic socio-demographical information such as age and sex. So if you would like to collaborate in this project, just feel free to drop me a line, and I would be happy to discuss. With that, I would like to thank you for your attention and to very much thank my collaborators in the Research Center Jülich and the University of Düsseldorf, in particular my colleague Somaye Maliki, who has done much of the metabolic connectivity parcellation work, which I presented today. She did a lot of sensitivity analysis and really went deep into the data to do this work. So thank you very much to her, and thank you for your attention.
Sarah, thanks a lot. Do you hear me?
Yes, I hear you.
Excellent, excellent. Yeah, fantastic talk. Particular thanks for using the approach of molecular connectivity. So I will moderate the questions and answers session, so to say. Other questions from the audience, from our virtual audience, please go ahead. Don't hesitate. I will give time to warm up and ask the first question. Sarah, you showed a very good overlap between different white matter tracts of the hippocampus and this metabolic networks.
Mm-hmm.
Did it make sense, or does it make sense to use also this parcellation as seeds for structure for fiber tracking, for tractography, to see how, if this, let's say, secondary structural parcellation, based on metabolic connectivity, overlaps, or how well it overlaps with the known anatomy, structural connectivity of the hippocampus. Do you know what I mean?
Yes, that would make sense. That being said, as I explained in the introduction, I think the best parcellation for studying white matter tracts, a phenomenon influenced by white matter tract, or using seed region in the hippocampus, would be probably regions that are defined based on structural, so anatomical connectivity. From what I remember, there are not many studies which have parcellated the hippocampus based on structural connectivity. And the reason is that the way the hippocampus is embedded within white matter tracts makes it really complicated for tractography techniques, actually. They often fail within the hippocampus. So having fine connectivity profile for hippocampal voxel based on tractography derived from diffusion would be very challenging. I think that's the main reason. But as a proxy, as you say, we can maybe use the parcellation based on metabolic connectivity, or maybe based on structural covariance. This may also be relevant to use a parcellation based on structural covariance.
Okay, but you would stay within the same modality.
Ideally, yes, that would really be my suggestion.
Thank you. The other questions?
Yeah, I have a question. Hi, nice talk. I'm sorry, I was fighting with my computer desperately, and I missed about half of your talk. So my question is really a methods clarification. So I'm sorry, I tried very hard, but so your metabolic connectivity-based parcellation. If you know, I'm probably missed exactly this part. You computed correlations between the hippocampus and the rest of the brain, and then used some clustering or some gradient-based technique. And these correlations are there across people? Maybe just quickly to clarify.
Exactly. So we have a static FDG, and we do the correlation across a pool of cognitively healthy subjects.
Okay, great.
A follow-up question, also methodological, Sarah. I am of course aware of this k-means approach. Was it, Christian, by the way, clustering approach? Does it take into account only connectivity, or covariance information, or does it also take into account amplitude or signal intensity? If it could be, the reason why you found in the end also significant differences in glucose uptake within these 5 subregions.
No, it doesn't take at all into account intensity. What it takes is really for each voxel, the vector of, so the value of connectivity with or correlation with all other voxel in the brain. So here it's not voxel, it was parcel, but I should have so this is here this is another region, but this is exactly the same idea. You have the seed voxel and you have the other voxel in the brain, or parcels. For this metabolic connectivity-based parcellation, we use actually parcels and not voxels in the brain. And for each hippocampal voxel, we have a value that we have different value that reflect the correlation in metabolism with each other parcel of the brain. And this is the information that is used for clustering or factorization.
I see. So the amplitude is not considered here, I think.
No, at all.
Right. Yeah. I have also, if you don't mind, once we're talking methods, I always, and maybe I caught the tail end. I think you mentioned bootstrapping and in its sampling variability. I wonder, with k-means, I always wonder, because there's inherent stochastic variability, right? So submitting the same data, even from the same subjects, twice might give you a different result, right? How are you dealing with this? It's more for my benefit because I'm always struggling how
as far as I remember, we have different initiations. So we iterate with different initiation. And then this is also why we do this, because this can also vary a lot. The parcellation, if we do it at the subject level, so for metabolic connectivity, this is not at the subject level, this is done at the group level, but we know that at the subject level, it can vary a lot. So we expect that at the group level, it can also vary according to different samples. So this is why we use this bootstrapping. So we define different sample and then we average across sample to get a stable clustering. And we also have this information of how much it varies across bootstrap sample that we use for deciding which parcellation is the best cut, because we assume that a parcellation that is stable across different sample is closer to the ground truth than a parcellation that would vary a lot, a level of parcellation that would give completely different pattern across different samples.
Great.
So other questions? There is a question in the chat. Thanks for the talk. I have two questions. Yeah, you have used metabolic connectivity as a basis for hippocampal segmentation. How much was it different from functional connectivity segmentation, if there are any published studies?
So typically the functional connectivity parcellation of the hippocampus, I should have them somewhere. Let me try to go back to this. Where are they? No. So we have a range of parcellation here, but they are really cutting. Often they subdivide the hippocampus very much on the anterior posterior axis, the functional parcellation. So it's really, as we go across granularities, subdividing the hippocampus from anterior to posterior. And the most stable, robust model in functional parcellation of the hippocampus is this three-parted model, so with the head, body, and tail. We don't have this much medial lateral differentiation in functional parcellation. And maybe we'll go back to this, but I know it doesn't work that way. Let me just check something. I have lost the. Okay, it's here. I have to go that way, sorry. So we also compare this parcellation based on metabolic connectivity with cytoarchitecture subfield, and we had quite good correspondence with the quite good overlap with the cytoarchitecture subfield. This is why we decided to name them anterior subiculum versus posterior subiculum and anterior CA versus posterior CA. Yeah, so this was my point, basically, that functional parcellation is more really strongly subdividing along the anterior posterior axis, while here we have this quite early, we have this medial lateral differentiation. Maybe I can. So you actually see that at two cluster solution, we already had something that looks a bit remind the CA versus subiculum subdivision with metabolic connectivity-based parcellation. But with functional connectivity, we would typically have anterior versus posterior region, from my experience, when we split into two clusters. So what I mean is that functional connectivity is very much subdivide the hippocampus directly from the beginning along the anterior posterior axis, which will not be the case with metabolic connectivity.
Sarah, I see that the difference is actually not big, right, between different models, like 6 clusters and 2 clusters. The stability is quite similar, a bit lower on average.
Exactly.
The stability criterion, is it sufficient to, to select the most appropriate segmentation?
So in other work, we also use different metrics such as silhouette indices and some other typical metrics that are used in data science, I would say. But often they do not go all in the same direction. So we also have a more top-down driven approach in which we also think what would be a useful partition. Because if the left and right hippocampus are also completely different, for example, it is a bit difficult to explain and to study. Since we don't know whether this is an artifact because there might be more noise in one region than the other, or whether this is a true differentiation between the left and right hippocampus. So here we also took into account the symmetry between the left and the right. And also what we try to do is to go beyond, for example, two-cluster solution, which is often not very informative. We try to find a trade-off between a parcellation which has a certain level of partition, but it's not neither too high subdivision for such a small region.
Thank you.
I have a question. So, have you tried to do this parcellation on cortex, like in a larger spatial scales? Whether a PET will be works or better compared with both?
No, we haven't done it in the whole cortex. I don't know if they are as far parcellation based on metabolic connectivity on the whole cortex. People have used gradient recently, I'm quite sure. But whole cortex parcellation, not exactly. I have a collaborator who has done in the US some kind of factorization in order to have different component, let's say metabolic component, but this was not, as far as I remember, in purely healthy subject. It was in AD patient in order to, again, have just a map or a subdivision to study something in Alzheimer patient. So this was not in LC. Here, we really wanted to have a parcellation which reflect a subdivision in LC subject.
Yeah. So I think.
Sorry.
Go ahead. Go ahead.
No, I said I really enjoy the discussion about the metrics and the clustering, and I think also maybe not just relying on data science, but on neurobiological meaning or concordance with the cytoarchitecture, might help beyond just silhouette scores or all these other things. And my actual question is, if you thought about doing this in diseased people, and maybe you will at some point, maybe you won't. Would you think that the parcellation looks much different or disturbed or wouldn't show such good concordance with the cytoarchitecture? Say you took early Alzheimer's disease people, for instance.
Yeah, I think it could look different. It really depends on the way the disease would affect the metabolism in this region or in other region. So either the disease affect more some subregion than other, then we would have a similar parcellation, or it actually tend to result in less segregation, which we often found in many diseases that there are actually less segregation, and therefore we have a different parcellation. So regions are not subdivided; they just are uniform anymore. So there are these two type of hypotheses that can be considered, but we haven't done it in disease. As far as I think, the number of patients was not that high in that time in ADNI, and also the quality was maybe not that high either, from what I remember.
Thank you, Sarah. There was a second question in the chat regarding resolution, which may I extend a little bit, because hippocampus is a sort of unthankful structure in nuclear medicine. I will explain why. Because it is has a very complex shape, configuration. It is close to the skull base, and there has been for a long time a discussion why we don't see any hypermetabolism in the hippocampus in Alzheimer's disease. So in patients with Alzheimer's disease, so we see hypermetabolism in the parietal cortex and the posterior cingulate cortex, but not in the hippocampus. And then there were some explanations. While we know that the hippocampus, as part of the mesial temporal complex, is one of the earlier structures affected in Alzheimer's disease, and there were explanations like incorrect or inaccurate spatial normalization, or sprouting of hippocampal neurons. Yeah, so this is a sort of mechanism of pseudocompensation, which would increase the metabolism potentially, and therefore the region would not appear hypermetabolic as compared to the neocortex. And yeah, and of course, PET has a limited resolution, and here you go quite, yeah, not ambitiously, but quite challenges resolution issue where you, I don't remember if you use parcels or voxels to examine this metabolic connectivity in the hippocampus. So what do you think, how, yeah, reliable is this voxel-based correlation in such a complex structure, and in particular related to the Christian? Yeah, if we work with patients where we know there is an atrophy of the hippocampus, yeah, would this segmentation still more or less accurate? You know what I mean? So it's
that's a very good question. We struggled a lot with that when we did that study. Actually, we Sommeyer did spend at least more than 1 year in revising the manuscript after submission. So what we did is that we selected different sample within EDNI, so doing some kind of sensitivity analysis to ensure that, because EDNI has multiple sites and some have better quality, better resolution than others. So there were a small sample in which the quality, the resolution was really better. And so we made sure that we could replicate the parcellation in these different sample, which differ based on the quality and the resolution. And also from my experience, this might be a bit surprising, but actually, even if the voxel are not really accurate, you have some kind of smoothing effect with this small spatial inaccuracies that at the end does not affect so much the clustering, because it somehow gets the overage. So if the voxel are slightly misplaced or changing across subject, it does introduce a little bit of variation, but you still get to the core subdivision by overaging across subject and across sample. So it actually works well. That being said, I wouldn't go into I wouldn't be very confident in going in very atrophied brains for such parcellation based on metabolism, which is typically not high resolution. And for other part of the brain, I would also be very careful. As you say, it may depend where you are exactly in the brain. As I say, for parcellation based on structural connectivity, for example, it's typically fail with standard tractography approach within the hippocampus. So this really depends the region you are looking at and the features. Yeah.
Okay, thank you, Sarah. Thank you. I think it would be still very interesting for us as a group, because we pay much attention to validation of this molecular connectivity, and your networks based on metabolic connectivity, hippocampal networks, really overlap very well with structural connectivity, which shows somehow it makes sense, this connectivity. If it could be somehow quantified, this would yeah, support this approach. Yeah. Other questions?
Yeah, I have a follow-up question. Basically, is that you mentioned at the beginning that it's complicated how to define regions, but like for us, is that usually the goal is, for example, we define regions of interest for the whole brain, and then we perform graph theory analysis or brain behavior correlation or predictions. So like, what kind of a population do you recommend? And you suggest that, for example, different modalities maybe use different populations, but sometimes we want to compare them, right? And also, I see a lot of study using AAL, for example, especially in PET. But what is your recommendation for choosing parcellations?
So if you, that's a very good question, because we also have this challenge. So if we focus on one modality for association with behavior, for example, as I say, we will really try to get a parcellation which has been based on the feature we study, and ideally not from the sample that we use for the association with behavior. So there should be a parcellation which is based on these brain marker or these brain features, but in a different sample or in a different data set. But what we want to compare across modality, this is the big question. What we do is that we use a functional parcellation, because we think that, so typically for example, the Schaeffer Atlas from Tomasio lab, because we are very much interested in behavior and clinical symptoms in clinical population, and therefore a functional parcellation is usually what make most sense. And the parcellation, anyway, based on metabolic connectivity, structural connectivity, functional connectivity, of course, but also structural covariance, often are grossly aligned with parcellation based on resting state functional connectivity. So this is typically the parcellation we will go for when we have to compare across modalities, because this is, most modalities are, most features are grossly aligned with this functional connectivity parcellation.
Great. The last chance for a question. If
Nicola, I think, has a question.
Yeah.
Hi, Nicola.
Hi, Sarah. Wonderful talk. Thank you so much. I've got a question concerning what parcellations to use. So when you use the Schaeffer parcellation, for example, there are different levels of granularity for that parcellation, and do you have a favorite level of granularity of the Schaeffer parcellation? And if yes, why would you choose, like, say, for example, the 400 versus the 1000 parcels version of the Schaeffer parcellation sheet?
So we actually have a very pragmatic approach. So for example, if we try to predict some behavioral scores from brain, we try different level and we see the one that works best. So this corresponds to the best partition to best capture the signal and the variability across participants, therefore for predicting behavior. But we also have other practical constraints. So if, for example, we work with more than 400 parcels, this means that we have more than 400 variables, and this become really high dimensional. And if we have a low number of participants, we end up with a higher number of variables than the number of participants. So we try to go to a trade-off. So usually if we have a relatively small sample, like 200-300 participants, we will go for a lower level partition, because it doesn't make much sense to have 1000 region if we have 100 participants, for example, for the type of statistic that we do.
I understand. Thank you. That's a nice clarification. Thank you.
Igor, I think you're muted. I don't know, it's on purpose. Yeah.
No. Thank you, Sarah. Thank you for the Hinweis. I encourage the audience to share your FGGP data with Sarah. I shared my own already.
Thank you very much.
And Sarah, I wish you much success with your new and very important project. And
thank you very much.
Wish everybody a nice weekend and join our MCOS series in the future. Thank you.
Yeah. Thank you very much for the invitation, and thank you very much for the discussion. It was really, really nice to talk together and discuss this work together. Have a good weekend. Bye-bye.
Thank you. Bye-bye.
Bye-bye.
Yeah, that was a really good talk. I really enjoyed it.
Thanks a lot. Bye.